Hi everyone. Welcome. So you just saw a brain and a little bit about the sensory system, but the other side of the story is the motor, which I'm sure you're all familiar with. If we just kind of take the same picture that Jim just showed you, everything to the left of that purple is the sensory areas, most of it anyway. And that separation between the orange and the purple is kind of that line where everything in front is, immediately in front is motor. Of course, there's a lot of other stuff like thinking as well. So we thought it would be kind of fun to focus on both sides of that story. So this activity is based on the SpikerBox from Backyard Brains .com. The guys were neuroscientists that developed this little box. You guys are going to get to use the box. So the whole issue about measuring motor function is oftentimes our motor function is something that's following a thought. So we think about moving. Sometimes we don't think, it's just voluntary. But oftentimes, if you ask me to pick up this picture, I'm thinking that I have to go pick up that picture. And somehow that translates into moving my arm and picking it up. And so a lot of the graphics here are from the Backyard Brains website, which comes with the SpikerBox, where you can go to the website and see all this stuff. And basically, this is just a cartoon outlining for those of you who, well, let me just stop for a second. How many of you teach biology? Chemistry? Physics? Okay, well, I threw a little of each in here. So for all of you that are teaching these different courses, you can get a sort of a flavor for how those particular subject matters feed into this kind of activity. And on this cartoon that came from the Backyard Brains site, basically it's just showing you the whole picture. That, this thought, which of course starts in your brain, is going to end up causing a muscle to move. And the neuronal information is going to travel from the brain down the spinal cord, and a little blow-up shows that there are neurons connecting with each other or forming a synapse in the spinal cord over there on the left. And then the motor neuron leaves the spinal cord and forms a synapse onto the muscle. At that synapse, the neurotransmitter is released, acetylcholine, for those of you in biology, and magic happens, right? The chemical acetylcholine converts the signal into an electrical one. And if you remember your biology from way back when, even if you're a chemistry or physics teacher, those neurons are going to transmit information, electrical signals, toward the terminal where that electrical activity causes the release of acetylcholine, shown by the little circles there, and that acetylcholine binds to acetylcholine receptors on the receiving cell. In this case, it's another neuron, but today it's going to be a muscle. Every one of your muscles is contracting because acetylcholine is being released at them. Okay? It's just a 3D picture from a film, an animated film that I made several years ago called Animated Neuroscience. And it shows the animation of this happening in kind of like 3D. It's kind of cool. These are the receptors. Similar to the kinds of receptors that Jim talked about, those proteins that sit on the cell membranes of the receiving cell, whether it's neuron or muscle. All right, so that was the biology. Chemistry and physics has to come into the story too. So in order for the acetylcholine chemical to convert the signal into an electrical one, we have to look at how the cell is set up, whether it's muscle or nerve. And that cell has a membrane, as you can see, with little purple lipids here, phospholipids. The sandwich, remember the sandwich, the bilayer. And it keeps your sodium ions on the outside, which are high, and potassium ions on the inside of the cell, which are high. And they're going to move in the direction of their concentration gradient if something lets them, like through a receptor that's got a hole in the middle, which we call ion channels, or through other little ways that they can sneak through, or a protein can take them back and forth. And so at rest, this is what it looks like, this nice distribution. So there are more positive charges on the outside than there are on the inside of the cell. And by the way, that distribution sets up for the physics people in the room a voltage difference, and that is at rest minus 70 millivolts. So when the acetylcholine comes along and binds to its receptor, it sets off a situation where ions move back and forth through that receptor, and it basically makes the outside of the cell more positive. The inside of the cell more positive, because ions are flowing through the receptors. And when that happens, these little plus signs on these ion channels, these are called sodium channels. So they are proteins that have a hole in the middle. They open up and only let sodium through. So they're called sodium channels, sodium ion channels. Those channels open when the membrane around them is kind of positive, which happened from the acetylcholine. And when that happens, it's called a voltage gate. The gate opens, and sodium ions now flow into the cell. And that is the key to what we call a sodium current, and leading to, when there's enough of them all in one place, an action potential to form. And we can follow that action potential with a tracing from a recording, and you guys are going to record them today on your own muscles. So this just shows you on the left when the sodium comes in through the sodium channel into the cell, and also that increased voltage, the positive voltage. So maybe the membrane's now at minus 20 or getting close to 0 millivolts. The sensor on the potassium channel then says, open up. And the potassium channel opens, and which way are the potassium ions flowing? Out or in? They're going to flow out, because potassium's higher on the inside normally than the outside. And so when they flow out, we start to get the downside of this curve. So sodium going in helps to increase this action potential curve, and as we return to rest, the potassium ions are flowing out of the cell. And then we have helper proteins like the sodium-potassium ATPase pump that helps restore everything back to normal so you can contract your muscle again and not be in tetanus, right? You don't want it to be forever. All right, so how about some real action potentials? We're going to grip and squeeze. So what we're going to do is show you just some things before we grip and squeeze of how you can actually measure your action potentials, or look at them, or even listen to them with the help of the SpikerBox. And then we're going to grip and squeeze and show you what happens when you have some larger muscles and you start doing something with them, like gripping a hand grip. And these are the muscles here that are important, these 3 muscles in that hand grip. And what happens if you keep gripping and you fatigue the muscle? So what you're going to see are an actual motor unit, which is the neuron releasing acetylcholine to depolarize the muscle and then allow the sodium ions to start flowing and the potassium ions to go out. And each one of those little blips is an action potential, just like what I showed you before in the big blow up. Okay? That's one motor unit firing. But if you have stronger than a weak contraction, then you get that same motor unit now firing at a faster frequency. So more times per second, which we call hertz, the number of times per second. And in addition, you start recruiting more motor units. And in your handout, you'll see kind of a nice picture you can look at later of how they've actually modeled this with recruiting more motor units to have a more moderate type of contraction. And then if we do something more intense, like really holding our hand squeezer real tight, For a tight contraction, you're going to get a lot of strong, high-frequency action potential firing at all random. And that's kind of what it will look like. So you're going to see kind of how these things look on your own arm. And then we can see, well, what happens if you keep holding that squeezer hand grip thing? And as you can see here from the picture, something's happening over about 70 seconds. The amplitude, it looks like, is getting smaller. Does everybody see that? And so what I've done on your handout is I have put that picture there, and if you were to do this with your students, you could have them print it out from their computers. We're not going to do that here today because we don't have all the equipment to do that, but I've given you an example of how they could pick 2 time points, or I've picked 2 time points for you if you want, or you can do it yourself. We do have one ruler back there. And you can measure the amplitude. The other thing you could measure, if we had the software to do it, I don't, but I've downloaded it on my computer, and you can do that too. It's in your handout. It's called Audacity. Backyard Brains website has the links to it as well. And that's where I got it from. And you can actually measure the frequency. You can slow it down enough. The software will tell you how many spikes per second you have during that time interval that you want to measure. So you can see whether the amplitude as well as the frequency are changing as your muscle gets tired. And then you can try it on different muscles to see if different muscles tire differently. So there's all kinds of questions you could throw out to the kids. Say, well, what if? And then the Backyard Brains website has some other kinds of activities with muscle fatigue that you could do as well, holding heavy things, stuff like that. But so we are looking at how the brain controls our muscle function. And we're actually measuring the electrical activity in our muscles, or the action potentials, using the SpikerBox. The SpikerBox is the invention of a couple of neuroscientists, and Tim is here, actually one of our inventors, from BackyardBrains.com. And he's made these boxes that allow teachers and students to actually measure the action potentials being generated in their muscles as they use them. So what we're doing is hooking up their muscles with some electrodes. We've got a certain type of electrode here that can measure the muscle activity of their fingers sideways here, or we have leads that we can put on our muscles in our arm and play
and you can actually use this
with a hand grip to measure not only the intense activity going on here, but how fast it fatigues. And we look at that information on our screen from the SpikerBox, the connection to the iPhone or the iPad. And if I were to put 2 of the electrodes on and actually hook myself up to this particular muscle and hold my ground, of course, and squeeze, you can see the activity of my muscle. And if I keep holding, that's going to decrease. So the students can play and hypothesize with which muscles are controlling which activity, and it's a wonderful device to learn about how the brain controls motor activity and how we can actually measure the individual changes of motor units in different muscles. So that's what we're doing today. So basically, each lead has to be,
well, you won't move them, but for anybody else that's going to put it on, you might want to put them a little closer together. Don't move it because the gel is now. Let's just see how it works. So you'll put one on each lead, and then this is your ground. So this can go on your ring, or you can hold it in your other hand. Okay, it just needs to be attached to your body somehow. In the meantime, you guys, you're going to be on the front end here. So you want, if you're going to move, some fingers, or your thumb and finger, you want your electrode.
You want on your ring?
Here or here. Yeah. So these are your action potentials. You can play with that's neat. You can play with the time frames. It's so cool. So move them this way. So there you go. Now if you move them up and down, not very good, right? That's because that's a different muscle doing that. It's up here. So you would have to put these up here if you want to measure up and down. So you can have the kids play with which muscles you need to record from in order to, understand what's driving the action potential. What I did is I bought one kit that was the EMG kit, and then the other kit I bought was just the standard one, so it didn't have all the other stuff with it. And then I bought just 100 of these because it was much cheaper. Yeah. You could probably get conduction gel pretty cheap anywhere. The complete kit is $149, I think, on their website, and the stripped down kit is $199. And I think if you buy in bulk, it's cheaper, and you could probably talk to Tim about that. But there's other activities you can do with this on the website. There's like 10 of them at least, 15 of them. There's all kinds of stuff you can do.